Okay, so we need to think about C4 cycle. That's where we left off. And so we're thinking about the idea of plants trying to minimize how often the PCO cycle is occurring. And so we talked about this last week, how in general, it's not a good idea to have the PCO cycle running continuously or super frequently. And if you are in hot, dry climates or really hot climates, this can be a problem because under those environmental conditions, the PCO cycle does kick in. And so what they're finding is that there's many plants that are native to these dry, hot conditions that are capable of running the C4 cycle. And so remember, they have a C3 cycle. All plants have a C3 cycle. It's just that they kind of tack on this additional C4 cycle and link it into the C3 cycle. And so it's a way for them to create a situation where that sloppy rubisco enzyme doesn't see a whole lot of oxygen. Instead, it sees lots of carbon dioxide around it. And as long as you can create that situation where rubisco is seeing way more CO2 than O2, then CO2 will outcompete oxygen and CO2 will come into the active site of rubisco regularly. So that's what these plants are trying to accomplish. Here we're looking at the C4 cycle kind of tacked on ahead of the C3. So when we look at this diagram, they're showing two different locations. They're showing two different cell types. You have the mesophyll or parenchyma cell, so maybe that's a spongy parenchyma cell in the leaf. And then you have the bundle sheath cells, which is those ring of cells that are around the vein. And so you're looking at how the C4 cycle actually starts in the spongy mesophyll or spongy parenchyma cell. It's coming in, and it doesn't react as rubisco right away. Instead, it's PEP, and the phosphoenolpyruvate and CO2 react in the active site of pepcase. And that enzyme is really good at discerning the difference between CO2 and O2. It only will take CO2 into its active site, which is good because now we can have the carbon dioxide and the PEP reacting. The first stable product is oxaloacetate, which then goes through a reaction to give you malate. Now, once malate is formed, that's good because it's the so-called, I like to think of it as a vector or a carrier of CO2. So CO2 is part of its structure. It will mobilize out of the parenchyma cell or mesophyll cell into the bundle sheath and deliver some CO2 to rubisco that's about to kick off the Calvin cycle. So you can see the way they have this drawn, the Calvin cycle is actually happening in the bundle sheath. You don't see the Calvin cycle being drawn as happening in the mesophyll or parenchyma cell, right? So that's why they say there's a spatial segregation of where things are happening in these C4 plants. So if everything's going okay, this C4 cycle will provide a mechanism where malate will keep coming into the bundle sheath cell, CO2 keeps getting cleaved off of it, and that CO2 gets fed to rubisco. And then the Calvin cycle can kick in and do its job of making sugars, recycling and regenerating some more RUBP to keep things going. Once the CO2 is cleaved off malate, you end up with pyruvate. And so now it's a cycle, C4 cycle. So now you have to get that initial molecule made again, that PEP. So that's the job here. Pyruvate needs to go back to the mesophyll cell and run a reaction where you can get more PEP. So you can keep the C4 cycle going round and round and round. But it's linked with the Calvin cycle in the sense that malate is delivering that CO2 into the bundle sheath where rubisco is located. Questions on the C4 cycle? think that's pretty much where we left off last Friday. So we were looking last Friday at this table where I kind of lay out where things are happening. So with the C4 cycle, there's essentially three parts to the C4 cycle. There's the carboxylation, which just means CO2 is coming in to the leaf and reacting with PEP. So that's happening in the mesophyll, spongy mesophyll cells. The decarboxylation just means that's where malate is getting cleaved, the CO2 is getting cleaved off with malate, and that's happening in the bundle sheath cell, BSC, bundle sheath cell. And that's where the C3 cycle is mostly cooking along. And then you have to get more PEP regenerated. So the regeneration step is to get that pyruvate pushed back into the mesophyll cells so that they can make PEP from that. So I put a little asterisk here by the C3 cycle because they've actually discovered that plants are really, C4 plants have really taken it to the extreme in the sense of how they run the C3 cycle to make sure rubisco does not see oxygen. So what they have discovered is that in the bundle sheath, CO2 is getting cleaved off, rubisco takes the CO2, and you have to remember that when you think about the first step, and let's go back here. When you think about the first step of the C3 cycle, we're looking at this idea where CO2 comes in, rubisco will then allow RUBP and CO2 to react, and you get your two identical molecules of PGA. So that's the first step. So that happens in the bundle sheath cell. Where'd it go? There it is. So that happens in the bundle sheath cell. So then that PGA gets made. Then what the plant does is it takes the PGA that it just made in the bundle sheath, it actually puts it out into the spongy mesophyll and says, please do the next phase for me, which is taking PGA and running that reaction to get you G3P. So there's this little tweak in the C3 cycle. Most of it takes place in the bundle sheath, but the second phase of the Calvin cycle actually takes place back in the spongy mesophyll cell. So they'll initially, that CO2 comes in the bundle sheath. Rubisco takes the CO2, RUBP, reacts it, gets the PGA. PGA gets sent out to the mesophyll, and the PGA will get converted to G3P, which is like this. So here's your PGA. This gets sent back to the mesophyll, and it gets phosphorylated and then reduced by NADPH to get you G3P. So that seemed a little weird. Why would a plant bother to split that phase of the Calvin cycle and have that happen in a physically different area in the mesophyll cells instead of the bundle sheath? So they looked at that more carefully, and what they realized is that when the plant is doing that, it's actually even better because when you take PGA and phosphorylate it and then reduce it to G3P, you're using ATP and NADPH to get that to happen. All right, just like here. Well, how does the plant get ATP and NADPH? They're getting that from the light reactions. Well, what else is getting created in the light reactions that we don't really talk too much about during the light reactions? Oxygen. So what's happening here is they're making sure that the bundle sheath cell really doesn't do much of the light reactions at all. The bundle sheath cells are more devoted to let's run the Calvin cycle. We need rubisco for the Calvin cycle. Let's make sure we're not creating a lot of oxygen in the bundle sheath. So the step of the Calvin cycle that needs the products of the light reaction, or at least most of it, that gets done back in the mesoscale cell. So again, they're spatially segregating a step of the Kelvin cycle that they know, it's ATP and NADPH. You need that. Well, let's put it out into the mesophyll. Let the mesophyll cells make all that ATP and NADPH through the light reactions. Let's allow the bundle sheaths to just focus on the Kelvin cycle. So again, the spatial segregation. And then once the G3P gets made, that comes back into the bundle sheath and they finish the Kelvin cycle. So again, it's this spatial segregation of what's happening with regard to the light reactions, the Kelvin cycle, making sure there's not a lot of oxygen production going on in the bundle sheath. So that means not much light reactions are going on in the bundle sheath, because that's how they get oxygen synthesis. So they do a really good job at making sure Rubisco does not see oxygen by flooding it with CO2 through the C4 cycle. It just makes sure Rubisco sees a ton of CO2. And then they minimize how much oxygen actually gets made in the bundle sheath by letting the mesophyll cells do the light reactions. So very slick way to make sure that you don't have Rubisco surrounded by oxygen. And so the CO2 will win. CO2 will come into the active site and you'll get Kelvin cycle cooking along all day long. So they've been very interested in the C4 cycle because it is it's a very good adaptation to hot climates. And they've noticed that there are at least 16 different families of plants that can do where the C4 cycle can be found in at least some of the species. It ranges from monocot angiosperms, dicot angiosperms, so it's not like it's just one or the other. Some of the more famous ones that have been studied, the families, Poaceae family, used to be called Gramineae family. The Cyperaceae family has been heavily studied, but 16 different families. I'm not going to list them all out. And so if you add it all up, it's about 1% of all known species, angiosperms, that can do the C4. In terms of agricultural crops that are important for human food production, corn, sugarcane, sorghum, some sedges, and millet. So some very important agricultural crops. Unfortunately, some so-called weeds are also famous for C4 cycle ability. So crabgrass, Russian thistle, pigweed. So these can show up in ag fields and in your lawn as well, and are considered kind of a pest species because they take up space and resources that you want your agricultural crop to have. So this can be a big problem. And so anytime you have so-called weeds that are capable of the C4 cycle, it's an extra dilemma for the grower to have to deal with. So C4 plants, if you look at where they're native to, where they evolved to live, we're thinking about hot, dry climates. There would be a period of rain, but there are also periods of drought. If you consider that kind of climate, C4 plants will outperform C3 plants, hands down, because they just can do photosynthesis that much better. They're efficient, quicker, carbon fixation, both the light and dark biochemical reactions, and that pepcase enzyme, because that's the first enzyme that interacts with CO2 coming in the C4 plants. It's just really good. It will only work with CO2, nothing else. It's very efficient. And so this C4 cycle has a super efficient pepcase enzyme, and it has that ability of delivering malate into the bundle sheath so that CO2 comes right off of it. And so Rubisco is just bathed in lots of CO2 all day long. So they're finding that C4 plants, what they can do is they can, even under drought conditions where they're going, oh boy, I'm experiencing drought. And one of the first reactions to that is the plant will start closing down its stomata to conserve water in its body. Well, with C4 plants, they can reduce their stomata so that their rate of CO2 intake slows down, and they still are really good at photosynthesis. So even though they have a slower intake of CO2 because of their smaller pore, they are still really good at concentrating the CO2 around Rubisco, making sure the PCO cycle doesn't kick in. So when you do these careful experimental studies, they are showing that C4 plants definitely outpace the C3 plants under these conditions. So they say roughly, at the 25 degrees Celsius to 35 degrees Celsius, in that range of temperature that the plants are experiencing, C4 plants are still capable of very efficient photosynthesis and sugar production. On the other hand, if you compare that to C3 plants are adapted to cooler temps. They say 20 to 25 degrees Celsius is where they can be optimized for nice, efficient photosynthesis. You push them above 25 and they start to suffer. The PCO cycle starts to kick in. So C4 plants definitely have the edge under warmer conditions. So 25 degrees is, that's a nice, comfortable room temp, a little bit more than what we're experiencing in this room. And then 98.6, that's 37 degrees. So we're looking in the 90s. Plants are able to handle that quite well. C3 plants, on the other hand, they didn't evolve to live in those areas. So they just have a straight ahead C3 cycle. Okay, so and I think I mentioned last week, they are looking at trying to engineer plants that just do a C3 cycle to incorporate a C4 cycle. They're trying to genetically modify, and they're using a lot of synthetic biology techniques to try to get the C4 cycle to operate in C3 plants. And they're making headway, but they haven't succeeded yet. I think they will. It's just going to take a little time. One more I need to talk to you about is this one, crassulation, acid metabolism, big mouthful. Luckily, it abbreviates down to CAM. So it was initially discovered in the family Crassulaceae, so that's why they named it this way. Unfortunately, they named it before they realized that this pathway is actually found outside of Crassulaceae. It's not just this family, but the name stuck, so they kept it. So when we think about CAM plants, we're going a little bit more extreme than the C4 plants. We're thinking about desert environments now. Really extreme dry, hot conditions, long periods of drought. Now, these plants are amazing. So CAM plants, a lot of succulent plants will fit into this ability to do CAM. So you might have some houseplants that you have in your apartment or home that are doing this. We'll talk a little bit more about that. Again, they have a C3 cycle. It's not like they don't have that. All plants have a C3. It's just that they've tacked on this CAM pathway and linked it into the C3. And again, it's the big payoff, especially for CAM plants, is it greatly reduces water loss from their body. So their ability to conserve water is impressive. And this CAM pathway also allows them to make sure that Rubisco is just seeing a lot of CO2 and not much oxygen. So it's concentrating the CO2 around Rubisco. Okay, so C4 plants, we said it was a spatial or physical segregation of where things are happening. CAM plants, it's temporal, it's time-based. So if you can imagine yourself being a giant saguaro cactus in the desert of Nevada, and you experience day temperatures, blazing sun, not a cloud in the sky, well over 100 degrees soon after sunrise, all day long periods of drought. Would you want to open your stomata? Unless you have a death wish, I don't think you'd want to open your stomata during the day. So they don't. They're odd in this way. they will actually open their stomata at night. If you've ever camped in the desert at night, you know that temperatures can plummet dramatically from over 100 degrees way down, and the humidity levels come up a little bit during night. And so it would make a whole lot more sense to open your stomata under those conditions where you're not going to lose as much water out of your body. So at night, that's what they do. They open up their stomata and they start taking in that CO2 all night long. But they have to store it because remember, photosynthesis isn't happening at night. The Calvin cycle is not happening at night. So they have to figure out a way to store it. And then when the sun hits their body in the morning, the Calvin cycle kicks in and they can feed that CO2 into the Calvin cycle. So that's the decarboxylation idea, where you get CO2 cleaved off and fed into the Calvin cycle. Fixation just means you're making sugars from this, okay? So now the beauty of the CAM cycle, especially for you having to learn it, it's a lot of the same words and same pathways that we just talked about. It's just that these plants put it in a day-night rhythm rather than physically separating it. They just do certain things at certain times. So a lot of the same molecules are involved, same enzymes. So you don't have to learn a brand new pathway, really. So let's look at that. So here on the left is the dark, what's happening at night. And then on the right is during the day. So here you can see at night, the pores are open, the stomata are open. CO2 is being brought in. And what you see here is that it's PEP, phosphoenolpyruvate, and CO2 reacting, just like what we saw with the C4 cycle. Same enzyme, pepcase, so that's nice. Same exact enzyme. You get oxaloacetate again, just like what we saw with C4. You get malate again. And then what they do is they stuff malate into their vacuole. That's how they're storing CO2. It's in the form of malate. So all night long, malate gets stuffed into the vacuole. Malate is an acid. You can actually put a pH, one of those microelectrodes into the vacuole of a cactus at night, and you can see the pH just dropping, dropping, dropping all night long because all that malic acid is coming in. So all night long that's happening. And then when the sun hits the body of the CAM plant, they have to mobilize that malate out of the vacuole. So you see here the malic acid, malate coming out into the cytoplasm. It gets decarboxylated, CO2 gets cleaved off, and that will get fed right into the Calvin cycle in the chloroplast. You end up with pyruvate again, just like what we saw before on the C4. And so then pyruvate, it just says arrow, 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 arrow. And then you end up with starch. So that's a difference. With CAM plants, what you see, in order to get phosphoenolpyruvate, you have to break down starch. So that's different from the C4. All right, so it's the chloroplast's job is to store starch. So that's where you're going to see conversion of starch to get phosphoenolpyruvate. And then when you need to get more phosphoenolpyruvate, really what they're doing is they're getting pyruvate built up into starch again. And then you'll get starch getting broken down a little bit to give you phosphoenolpyruvate. So it's a pathway, not a cycle, like what we saw with C4. So that's really the only major difference between them, is that you start with starch and you end with starch in a way. It's not really a full cycle. So you have all of this going on in the same cell. So you don't have bundle sheath cells doing one thing and mesophyll cells doing another. This is all happening in the same cell. So all the mesophyll cells of the, well, a lot of cacti, is it cacti? I always forget, cacti? Cactuses? Cacti? I can never get that straight in my head. Anyway, I'm going to stick with cacti. A lot of them don't have leaves, right? I mean, think about all the different, especially if you have houseplants. If they do have leaves, they're usually highly modified. So it's a lot of stem tissue, especially if you think about the beautiful giant saguaro cacti. It's really the stem tissue. Okay, so in words, what we just saw visually with the figure is the CO2 and PEP reacting again, just like what we saw with the C4. You get your oxaloacetate, gets converted to malate. That enzyme, that peptase enzyme is taking care of that initial step. And then you don't need to memorize the NAD plus malate dehydrogenase. That's creating the malate. You don't need to know that, but anyways, malate gets made and it gets stuffed into the vacuole all night long, which makes a whole lot more sense if you're trying to conserve water, which desert plants need to do. All right, so what we just described in words here, that's what's happening here in this diagram. You have that PEP, CO2, peptase, gets you oxaloacetate, malate into the vacuole. Then during the day, you got to get that malate mobilized out of the vacuole, get it into the cytoplasm, and there's the enzyme to cleave off the CO2. That CO2 then gets fed right into the Calvin cycle for the fixation of CO2. And then pyruvate needs to get converted back to starch in the chloroplast. So all of the cells in the leaves or stems of the desert plants will be doing this. There's no difference in physical location here. It's just what happens during the day and what happens during the night. So here you see the vacuole again with all the malate coming. The malate comes out, CO2 gets cleaved off, fed into the Calvin cycle where rubisco is sitting, and then the pyruvate gets converted to starch. So a lot of the same enzymes and molecules as what we saw with the C4. So a lot of analogies there. Now, CAM plants have some added flexibility in the sense of some, not all CAM plants, and insofar it's a minority. A minority of CAM plants have the ability to, under the right conditions, they can say, you know what, I'm not going to run the CAM pathway. I'll just stick with C3. I'll just do that. And so they would need to do that where it would only make sense to do that if they could risk opening their stomata during the day. That's the only way you're going to get the C3 cooking along is during the day. So if they're going to choose to do that, what do you think might have happened in their environment where they might say, I could risk opening up my stomata? What would happen? What would have to have happened for them to say, I can risk it. I can open my stomata. Rain, right? If they can open their stomata, that means they're risking water loss. So they must have a lot of water to play with if they're risking opening their stomata during the day. So the monsoon seasons that sweep through Nevada, for example, on a regular, well, hopefully on a fairly regular basis, they know when the monsoons come through. So right after that, you could see some CAM plants switching and just turning off the CAM pathway and just running the C3. Why would they do that? Well, and there is an extra cost, again, to running the CAM pathway. We already talked about that with the C4. There's an extra energy cost to running the C4. Same with the CAM. There's an extra energy cost and just resources too to run both the C4 and CAM. That's just more resources you have to devote to those pathways. So if you are in a situation where you could turn off the CAM, just hold off on that until it's absolutely needed, you could save yourself some energy and resources that you could put elsewhere. And so definitely they will do that under abundant water supply. And again, not all CAM plants can do this. There's relatively few so far that have been discovered to do this. But I always hedge my bets, I guess, when I have a lot of CAM plants in my bedroom. next to the window there, because I want them to have their stomata open at night, because then they're taking in the CO2 that we're exhaling all night long. And so they're actually cleaning the air quality of your room. And so if you were going to pick a plant to have in your bedroom, try to pick cam plants. And the trick there is not to overwater them. If you overwater them, if they're facultative, they'll stop cleaning your air at night because they'll have their stomata closed at night. So I always say that I'm mean to my plants, my cam plants, because I don't water them regularly. I want them to open their stomata at night. So something to keep in mind, but they will take in that CO2 that you're exhaling, especially if you are lucky enough to spend 8 hours sleeping or longer. It's nice to have some cam plants in your habitat so that they can clean up your air quality. Plants can do more than take in CO2 though. They can clean your air in a variety of ways. If I have time to squeeze that into a short little lecture sometime this semester, I'll talk to you a little bit about that. You should have lots of plants in your living environment because they really do clean up the air quality. So Crassulaceae family certainly do, there's many genera in that family that will do cam. But unfortunately, they named it after that family, and then they discovered, oh wait, there's more families that can do this. So there's lots of others. It's scattered, and so it's not like it's unique to one family of plants. And when you look at all the different species in each family, it's not a guarantee that all of them will do cam. So it's really a hodgepodge of what plants can really do this. So in one family of plants, you'll find plants that just do C3, and then you'll find some that do cam and some that do C4. So it's all over the place. Some that you may be more familiar with: cacti, euphorbias, pineapple, huge crop. Vanilla, again, that's not really food, but it is for, flavoring. Vanilla is used tremendously in flavoring foods. What do we get from agave? Oh, good, you still know that. You're awake. Yeah, tequila. And so that's probably $1 billion industry in and of itself. And so again, there are cam plants. Orchids, again, that's $1 billion industry. There are a lot of people who are crazy about orchids, and almost to the point of fanatics about it. So big industry. Did you know that vanilla is actually an orchid? We get vanilla from an orchid. It's the only orchid that we get something to do with food from. So food flavoring, I guess. It's an orchid, it's a vining orchid plant that's native to tropical areas. Cam can also be found in more primitive plants, in the gymnosperms. So that's odd to think about, but so that makes you wonder, like, okay, when did this evolve? How did that all happen? And so hang on to that thought. But when you look at the different C3, cam, and C4, and this is rough ranges of water loss. If you're a cam plant, water loss per gram of CO2 that's brought in, 50 to 100 grams. C4, it's 250 to 300 grams. And then C3 is a whopping 400 to 500 grams of water lost per gram of CO2 being taken in. So you can see by far cam plants win. They can conserve water really, really well. And then when you think about where are these different pathways showing up in the evolutionary timeline of the plants, started out with C3, and then they think the cam pathway actually evolved first before the C4. C3 came first. All plants can do C3, but then they think cam came next, because you can see cam pathways being run in some gymnosperms. Some funky, weird ones. Not like none of the gymnosperms you see around here can do that. And then the C4 cycle arose last. Okay, so what we're looking at is this the C4 and CAM evolved in plants that are adapted or native to areas that are more challenging in terms of being dry or super hot. And so they had to evolve mechanisms to allow them to persist and thrive in those areas. All of these cycles, though, the C3, the CAM, the C4, what we're really zooming in on is the ability of the Calvin cycle to keep running so that you can get sugar production ultimately. So, you know, with the Calvin cycle, we talk about how you get that G3P pool of molecules made, and then, you know, 5 out of 6 get recycled for RUBP, but one of them can get siphoned off, so to speak, and you get your sucrose. So that's great. And then the plant has to decide what to do with that sucrose, of course. And so depending on the situation that the plant's experiencing, maybe they have a lot of sucrose so they can store some up for a rainy day. They're like, okay, I'm going to save some, store it up as starch, have some reserves in case I encounter a tough time. I'll be able to call on the starch, break that down, and use it if I have to. So most do that. There are a few plants, especially there's a handful of agricultural plants that instead they don't make starch. They actually just stuff their sucrose into the vacuole as is. They don't convert it to starch. So there's a few plants that do that. The ag plants that are famous for that are oat plants, wheat, and barley. And so they don't really make significant starch. They just stuff everything in the sucrose into the vacuole. So that certainly, if they want to store up, they can do that. Or the plant, of course, might have to make some bunch of sucrose and send it or use it immediately, right? So you have to use some sucrose for your metabolic needs. You might have to export it so that the sink tissue can get that sucrose so they can do something with it too. All right, so we've talked about these ideas. I'm just refreshing your memory that there's a reason we're talking so much about the C3 or Calvin cycle. And so you really have these options of store it up, use it for metabolism, use it for maintenance or building biomass. But what we're thinking about here is going back to this. Where'd it go? Here. Going back to this one. So we're talking about once the G3P gets made, the last one on the drawing there says export. That's what we're talking about for sugar production. You get that exported out to the cytoplasm and you get sugar production. And then in this drawing, they're saying 5 out of the 6 G3P are actually used to do the regeneration step to get you more RUBP to keep the Calvin cycle running, but then 1 out of the 6 can be exported. So you have to imagine this happening over and over and over and over. So over time, you can get a fair amount of G3P getting exported for sucrose production. So once it gets out into the cytoplasm, that what you see happening here. All right, so here you see the chloroplast in green up top. Here's the Calvin cycle, and they're saying triose phosphates. That's your G3P, your pool of G3P. They call it triose phosphates in this diagram. So what they're showing here is that you have the G3P, it'll get mobilized out actually as DHAP, which they're showing. Let me go back to this one. So here you can see G3P converted to DHAP and then exported out into the cytoplasm. But you see that there is a transport protein that's involved in that. And so if you follow what's going on here, you see the G3P comes out into the cytoplasm or cytosol. And then at the same time, they're trying to show that you get phosphate coming in. So this is a transport protein, transmembrane transport protein, and it's doing an antiport process. Something goes out as something comes in, so it's anti-directional, all right? So G3P comes out, inorganic phosphate comes in through the same transmembrane protein. So they call it the translocator, PI translocator. But basically what they're finding is it's a one-to-one relationship. As one triose phosphate comes out, one inorganic phosphate comes in. And so what they're saying, they've done enough studies on this, where that transmembrane protein right there, that is a control point for, allowing the G3P to come out or not. And so what they're finding is if you have a lot of inorganic phosphate building up in the cytoplasm, by the laws of concentration, that if you have a lot in the cytoplasm, relatively little in the chloroplast, it'll follow its concentration gradient and diffuse in, which will then allow the triose phosphate to diffuse out. So what they're finding is if you have a lot of inorganic phosphate in the cytoplasm, chances are you're going to have good export going on of the G3P. That'll keep getting exported out, and you'll get then sucrose. So what you see here in the lower half of the diagram is once that G3P comes out, then you see the pathway, and we're not going to get into this pathway. Just know that it's happening in the cytoplasm. You have to get G3P out of the chloroplast into the cytoplasm in order to get sucrose production. That's all I really want you to know about that. Okay. And then the other idea here is that inorganic phosphate, it's kind of controlling how much inorganic phosphates in the cytoplasm really is controlling how much G3P comes out. So that is a control point for kind of regulating what's going to happen to that G3P. Is it going to get exported right away or not? And so it really is dependent on this inorganic phosphate concentration. There are also, they've been studying all the different enzymes here, and they also know that those enzymes are regulated pretty tightly as well. I don't have time to get into that, but just be aware that's not the only way that export is controlled. So it makes sense when you think about it, that there has to be a pretty tight control on how much and how often G3P comes out to make sucrose. That has to be tightly controlled. Otherwise, the plant would be a hot mess if they didn't have control on that. So there are other enzymes in here that they know are controlled as well. So we're talking again about that G3P coming out, and a lot of it has to do with how much inorganic phosphate is in the cytoplasm. So here you can see if the triose phosphate does not come out, it stays in the chloroplast. And then if you follow the arrows, what you're seeing is some of that last, here, let me go to this one. So here you're seeing if you combine two of those G3Ps and make the 6 carbon, and then they show arrow, and then they have that box, and then you follow it all the way down to starch. So this section right here that my laser pointer is circling is being shown here in the chloroplast. So what they're showing here all the way to starch, that's what's happening to those 2 G3Ps is right here. And again, I'm not asking you to know the step-by-step of this. Just know that, if the plant's saying, you know what, I want to make some starch, that's what they're doing. They're taking 2 of those G3P molecules and they run a series of reactions in the chloroplast. It all stays in the chloroplast to get you starch. So starch, sucrose, it's an either or in that sense. It's either you're going to shunt the G3P that way towards starch, or if you have enough inorganic phosphate flowing in, that means some of the G3P comes out for sucrose. So it's kind of like an either or idea. The last series of slides in this slide set are a bunch of tables. I just put those in for study purposes. If it works with your brain and helps you learn the material, but I'm not, we went over this already to the level I want you to know it. Sometimes it helps students to see it in a table format, so I included it, but it's not something that you have to, it's stuff we've gone over. It's just presented in a slightly different way. And if your brain works with that, great, go ahead and use it. There's some slides that show the chemical structure as well. So if you like that, like learning it that way, then feel free to take advantage of these slides. But all the tables at the end, it's stuff we already went over. It's just presented in a table format. All right, so we've covered all of photosynthesis now. We've done the light reactions in detail, and now we've covered the C3 cycle in detail and C4 and CAM. So the next topic we're going to get into is going to be a shift away from this now. We're going to be getting into, well, first we got to get into signals. How do plants interpret their environment? How do they pay attention to signals? And then how do they act on that? So we'll get a little bit into that, kind of a general overview. And then from there, we're going to start talking about, well, how do plants interact with their environment, including other organisms? All right, so new slide set on Wednesday, but we'll stop here for today.